showcase of research findings and advancements in clinical medicine, public health, and health systems. And I'm Kim McAllister, and I'm a program officer from the National Institute of Environmental Health Sciences. And I'm facilitating this session with Dr. Jean Guile, who's from NHGRI. So our first talk will be a keynote talk by Dr. Judy Gichauya. And Judy is a multidisciplinary researcher. She's trained in both informatics and clinically active radiology. She is an associate professor at Emory University and working in interventional radiology and informatics. She is helping with the Open Data Science Platform component of DSI Africa, and her career has been focused on validating machine learning models for health in real clinical settings, exploring explainability, fairness, and a specific focus on how algorithms fail. She's also heavily invested in training of the next generation of data scientists through multiple high school programs and medical student and radiology AI trainee courses and activities. And she's actually mentored over 60 students now over the world. So thank you, Judy.
Thanks for the invitation. And so this is a little different from what I just talked about maybe less than 90 minutes ago. And I chose this and was guided by Rolanda. So if you don't like it, you can kind of blame her a little bit about, I want to talk about shortcuts and how they can cause bias. How many people are working with medical imaging, some form of medical imaging? Okay, how many people want to work with medical imaging? A little more? Okay. Well, this is not just a problem for medical imaging, as you will see in a few minutes. So let's get started. What's this? A shortcut? How many people use this shortcut? Okay, great. Okay. How many people use this other type of shortcut? Okay. How did you know about this shortcut? Maybe some bold person can shout so that I can. Huh? How did you know about this shortcut? You don't remember? I see Professor Shakun there saying doesn't remember. How did you know about this shortcut? How did you learn about this shortcut? Word of mouth? Like, okay, so people learn about whichever shortcut, this one or the next one, from word of mouth. They don't remember. Any other last comments about this shortcut? Yes? Okay, so if you're doing something that takes annoyingly long, then you look for a shortcut. Okay, so how many people have explored how to change shortcuts? Okay, why do you change the shortcut? Maybe somewhere here there's some hands. I know Professor Hash raised it. Which shortcut did you change and why did you change it?
Maybe I'm used to it, or maybe I previously did it in another application or something like that for some reason.
Okay, so you're used to it, or you just like a different type of key. How many have changed it because their keyboard lost a key? Nobody? Oh, thanks for joining me. Okay, so some people have changed it because you've lost a key. How many people moved from Windows to Mac or the other way around and had to really change it because it was driving them crazy? Okay, well, so this is half of the talk, and I just want to show you how these shortcuts appear in medical imaging, okay? So we're gonna describe shortcuts, we're gonna look at them from medical images, and then I'm gonna talk about shortcut learning, and then this impact on bias, okay? So the best paper to read this is this paper by Robert. And it's a really easy paper to read. And essentially, he shows that there are shortcuts that exist across all domains. For example, here if you say caption this image, most of the time the AI says, well, these are grazing sheep, and they're not sheep in that image, and it's just because the examples that the AI models have been shown tend to have grazing sheep. Here's an example. I'm a radiologist, so I'll show you radiology examples. Here is saying, tell me what pneumonia is, but the AI system learns that this radiographic marker here that just marks the left side is from the patients in the ICU, and those tend to have pneumonia, and guesses that there's pneumonia instead of trying to give you the correct prediction. So I'm going to, the first studies, I'm going to talk about more about shortcut learning from this paper, so that the next ones I can move quickly for us to understand. And I mean, for all of us to come to the same point. So if I show you this example, although I'll tell you that I did see the zoo here in Mauritius, and there's a camel that has green grass around it. So, but that's very odd because most of the time you'll have the cows will have green and the camels will have sand, okay? And so if you use that camel in Mauritius for your AI model, it may fail just because that's not the example that it's used to. So there are a few terminologies. I'm not going to be very technical for this talk. You start off with an informative features when you're training your AI model. And if you are not very judicious with your training, then you have this overfitting. The people who are in the data bootcamp had this word used a lot, where you cram, you don't learn anything new, you just memorize what the data is saying. And then you have these shortcut features. So it's not really cramming. They are helpful, as all of us have realized about Control V, C, Control V. But but what you're trying to do is intended features. And I want to say that we don't really understand how to harness, these shortcut features and make them make sense. So when the shortcut learning comes from, it becomes when you have this balance of the spurious strengths, in this case, the background versus the invariant strength, and the spurious strength is stronger than the invariant strength, and so you end up with a shortcut. Simply said, if you have your examples are all of cows with green grass, then the background becomes the most important feature more than learning the animal that is in the image. And so you can either have, because of the dataset bias, right? So I have not seen a cow near on the beach. So if you present this type of information and sort of the association, that can be a problem. But also, the convolutional neural network are trained by sort of, looking at shapes and lines. So if you look at the example that shows you how deep learning models learn, you're going to see that it's looking at edges. And if I can convince you, then here, the model is going to look at the edges and tell you this is an elephant. But really, most of us, I hope, can tell that this is a cat because you're also looking at the shape and the correlation of things that can happen. So the way we train our models today is not very respectful of that. And so this is why, for humans, these are same categories. The school bus, they're different, they're grainy, but for deep learning models, these tend to be very different, classifications versus this, which are same categories for us, but the model is looking at the guitar string and then starts to classify this as the same. And it's because of how we train our AI models today that this is a problem. And so, they can occur everywhere. So in text, if you start to say we're in this era of bots and large language models, and just by changing the demographics, this is a very common example. You see differences in where the patient is referred. If you say, show me a picture of a doctor, they tend to be very characteristic, ethnicities and sex. And in case, even for games that the model can learn, if I don't move, then I can hack the game. And so it's called reward hacking. And you can have shortcuts pretty much everywhere. So not only in medical images. And so how we test for them is that you have to really understand results carefully. And this is like an area I'm very fascinated with. And so I've actually moved. It's more important, and I want to emphasize this, so that there's no small data data set from Africa. And it's going to be more important to have diverse testing data set than training data set for us to understand shortcuts. It's not because you need to test and test and test and see where things fail. And then this example cannot be random or too outrageous. It has to be within the medical domain. And then some people are trying to remove some examples so that to see that if that can work for testing. And so they're testing technical components and non-technical components, trying to see what's the difference between cats and dogs when it comes to medical imaging, thinking about fairness, and also thinking about how to interpret the results. Here, I like to tell my students that when you train a model and you have performance of anything AUC of 0.99, that's a suggestion of an error and something is wrong. And so you should not be celebrating that. And these are the ways you can start to audit and investigate that. So assuming that all of us move from the example and have a sense of what shortcuts are, let me use medical images to show you examples. So I briefly talked about this for pneumonia prediction. This work compared 3 data sets and just said, tell us what pneumonia is. And then they use these saliency maps to point, this is what the model is looking at, and it's clear, it's just looking at the radiographic markers and then guessing these patients are more sicker, they probably have pneumonia. And maybe this is why we didn't see any COVID work really do well for chest x-rays to detect COVID. And this is not easy. So we have a chest x-ray data set at Emory we've been working on for almost 4 years. And when we just looked at the radiographic markers, they're everywhere. There are different types. They are facing different ways. They are, some are very radiodense, very bright, some are not bright. And I can tell you, even subsequent work we're doing now, it's almost that if everyone looked at their shots, and I'm wearing this very nice swag from DSI Africa, if you have a button, the model can learn what type of button you have. It has nothing to do with your disease prediction, but if your EKG leads are very specific to the unit the patient is admitted, then it's easy for the model to learn, these patients tend to be transplant patients. These are the diseases that they have, and they do not always translate to the actual disease that the model has. Here's another example for pneumothorax. Pneumothorax just means you have free air around your lung. We treat this using a chest tube where we put a tube to suck out the air. And you see variable performance of the model when you have with a chest tube and no chest tube. Even if you've never read these graphs, one of them looks closer to the randomness versus the other one. And you find that the model just learns what the chest tube is, performs very well for the chest tubes. In the clinical world, that's not the important case, because if you have a chest tube, you've already gotten the treatment you deserve more than the patients who don't have chest tubes. And those are the ones that the AI model doesn't work well for. Here's an example for dermatology. And here, unfortunately, it's not unfortunate, it's just that their benign lesions had these patches, these just markers. And the model just learned that, well, if you have a patch, you don't have to look at the lesion, you're probably benign. And guesses that and doesn't learn the disease versus the images that don't have a patch and those are malignant. We know that there's some data sets from ophthalmology. Here is an example of retinal scans. These ones are taken from a tabletop camera. They usually have this artifact. If you've gone to the eye doctor, they have you lean in and take those pictures. This artifact is very difficult to remove. And that's it. That's all the model needs to figure out what patterns of diseases are being seen in your population. And so, here's a really good example using public data sets around COVID, showing the complexity of how people merge data sets. And this is not unusual. When you don't have enough data sets, you go and pick another negative data set and another positive, you mix them, and you say, this is negative, this is positive. This is what the authors did. And they show very good performance on the internal test, almost 0.99, but when you test the model on anything external, the performance drops. And they do one more interesting thing where they look at generative networks, where you say, show me what make a patient who's positive, negative, and the other way around. And they start to see that the model is just following the radiographic marker, the R, when you move across the patients. And more interestingly, they look at could you change the last layer of the model and tell me something else about the patient? And so could you tell me the projection of the image and the patient's sex? And here they have perfect performance on both the internal and the external test, showing that just because you don't provide information in the model, it's going to use whatever pattern it's figured out to be able to do its prediction. Here is a more detailed example of hip fractures. This one shows you that there's some patient characteristics, there are many metadata around the images, there are image characteristics, and they do one thing, which is to say, show me, can you predict the scanner model? Can you predict the scanner manufacturer? Things that are not relevant to your disease. And you can see for the scanner model, it's near perfect. It can tell you exactly what type of camera was used to acquire the image. And so it encodes these type of things, including how long you waited, what's your BMI, and what's your age. And those things then tend to be used for prediction. Here's an example from the NIH dataset for knee radiographs. There are many acquisitions. And the reason I show you is that I've talked a lot about the radiographic markers. They're everywhere. And this is data from 5 sites. And so they train a model that shows you can identify the hospital, ABCD, not the name, where the data came from with pretty good ability. But when you look at where the model is looking at, it's not necessarily what the humans look at. So shortcuts are not always what the radiologist is going to see and take home. And so we're seeing a lot of work on foundation models, and these are just data sets that are bigger data sets. And when you look and audit them, we see that they can encode the same characteristics, sex, race, social constructs in their models, and they can have them propagated downstream. Finally, you look at segmentation models. This just means you're drawing a circle. This is the gold standard for radiology, because when you draw something, the radiologist can see it and say, okay, I see what you're doing. Here is an ultrasound of a neonate. We measure their bones of the femur to tell us how, what's the age of the ultrasound, of the baby on ultrasound. And when you remove the caliper, the model starts to just land on the caliper and estimates around the lesion. But when you remove the caliper, the segmentation fails. And just more importantly is that all the data sets, so these people go back and sort of start to shift the location of this lesion, and they show that the lesions that are farthest away from the center, have poor performance. But if you look at this 4 data sets already in the public domain, the abnormal lesion tends to be very centered in the image, telling you that that's what the model is going to learn. It's going to learn, oh, the malignant image is just always center, and that's it. It doesn't learn the cancer or the abnormality on the image. And so if you're far away from the center, they show that your prediction performance drops. So thinking about those examples, how do they impact bias? And one of our most famous works is this work where we showed, when I'm on the continent, I like to describe that if you gave me your chest x-ray, I could tell if you're Kenyan, Ghanaian, Nigerian, which is ridiculous. And but you can tell that with a very good ability. And this is a picture of many, many tribe representation in one of the Senegal museums. But you could say, you could calculate many things. You could tell me my diseases, you could tell me how I'm aging, how much I'm going to spend in the healthcare system. And I even give my example. This is not my chest x-ray, but you could say I'm Kenyan, I'm female, I'm aging faster than my actual age, I have COPD, I'm spending a lot of money, but we don't know why. And when we look at the performance of the models, and when you look at out of distribution and in distribution testing across 4 characteristics, we find that the models that encode demographics the most are the most biased. And the models that say you're very Kenyan are the most biased. But this is not a biologic difference, it's just something, but we don't know why that happens. And it's very difficult for us to remove those characteristics, because when we remove one, it goes to another one. So when I think about shortcut learning and in the context of Africa, a lot of people, are very saying this is it. This is the AI for Africa. I actually think it's an opportunity because we don't have a lot of compute, a lot of data sets, that we can use our small data sets because they tend to be very outliers. They tend to be mainly abnormal, not normal. And we can actually start to think, how do we really train computer vision models? And this can happen with tabular data. It can happen with any data. For us to think about all these are possible topics for all the students in the room that we need to work on for us to make sure that the shortcuts is not what the model learns. And when we say this is TB, that it really is TB and not because you're learning a specific way or specific pattern that the model the images are acquired. So here's a snapshot of my group in Emory, and I'm happy to take any questions. Questions or not this one? Awesome.
Okay, any questions?
Thank you so much for the talk. It was great. What do you think are some of the things we can do to somewhat systematically unpack some of the bias that's happening? In traditional epidemiology, there are ways to quantify bias. What do you think we should be looking towards to try and unpack that bias and then start addressing it through better, more robust representative data sources?
Yeah. So this is something that I've spent a little bit of thought, and also my opinion has changed a lot. And the first thing is that we need to test, but we don't have good test beds. But testing is actually easy for people. If you think about the computers we have today, they're pretty good. You can do testing, you don't need GPUs, you just need a small subset of data. And I think we don't know where models fail. That's the first thing, right? So we always, your students always, they're afraid to come tell you the model is not working well. They want to get 0.99, but we should be encouraging them to show us what happened from 0.5 to 0.99. And we don't have places where we can publish model failures, right? And when we think about, so for example, if you think about all the work I've shown, this is because of testing. We don't have a systematic way of auditing. And so it doesn't matter what you train on. If you're not testing on diverse data sets and figuring out, why do I have these false positives? Why do I have, this subgroup of patients that's not working? Then, you're not going to move forward. Now, I will say most of us in the continent, at least the sub-Saharan portion, we wouldn't even identify as being Black. Like I identify being Kikuyu. There are all these labels that we need to move beyond, and approaching it from a failure mode is also interesting because it allows us to say this subgroup of patients, the model doesn't work well for, and then we can move to something else. But and that can also be reverse innovation, where like in the US, all we do is like black, white, and then we stop there,
Yeah. Hi, Judy. It was nice talk. So, yes, there's a need to unlock bias and models, but I don't have a lot of experience working on image data than genomic. But from genomic perspective, we know most of bias come on heterogeneity of the data, and all those multimodals of from multi-omics and all those layers that comes in there. But it's always the question, I don't know, I just ask for your experience on working on an image data set. So it's a question of who to blame. Is the data or is the model to blame?
I think
So that is always the question myself I always ask when I do the model. So there's one part of the data that you did give a nice talk in the morning. I did have a certain question, but that is the point there today is to try the best in the model perspective to do AI, but also to do learning. It's kind of you bring data sciences component and AI together, and the so-called machine intelligence. So that is, I don't know, what is your point of view on that, or what is your experience on resolving that issue of whether to blame the data or to blame the model, and you in the between, how you resolve it in your context of imaging research?
So maybe next time we can have a debate of this very controversial question, right? The computer scientists say it's the data. The public health doctors, everyone else says it's the model. And so I feel that, the first thing is that we do not have a universal definition of fairness, right? So you can have a biased model that is deployed with a health equity in lens. So you could say, it turns out that for these patients, my model doesn't work well. And you focus resources on those patients and you have a better outcome for them. So the first challenge is that we don't have data sets that are linked with an outcome. So when you think about sort of the data sets, there's no clean data set. So if we say we're going to wait for the perfect data set, there's no perfect data set. If you build that perfect model, it's never going to work in the real world setting. And now we're getting into a new era of models as a data set. So the foundation models, right? So those models are not just models, they are data set. And so I think that line is going to be blurred because they encode the characteristics of the data set they were trained on, but you don't have a way to audit them. And so I think the way I approach this is that this is a very gray area that we need to initially sort of put a lot of work and just different views for us to figure out what works and what doesn't work. And that means, for example, I'm someone who works on bias, but in the real world setting for medical imaging, we don't see a difference between black and white in model performance away from research, but we see the models fail for specific subgroup of patients, and those tend to even be more interesting. So today, I would use the tools for hypothesis generation, because you can look at big patterns of data and say, why does this data set, what's wrong with this cohort? And then when you go back and look at that cohort, I think from like a true traditional science way of doing research, you can ask very interesting questions in that manner. Oh. There's no good question that starts with okay.
Okay. Thank you for that very nice and brilliant talk. I don't know if I missed that, but when you have such bias, like in image analysis, and if you think about brain images, MRI brain images, so what we do normally is to do region of interest. So we segment, we extract a particular anatomy, and then we look at that. So the noise issue completely removed. That's one. Secondly, for CNN, there are CNN that have been modified, to be region specific. So you localize a particular, abnormality, and you look at that. So all bias is removed. So I don't know if you have thought about that, or maybe you mentioned it and I didn't capture. Thank you.
No, so I did not mention ways to remove bias. And number one, there's no way that you remove bias. This is just a fact. You think you remove bias, but when you go back to test your model, you find another problem. And so when it comes to segmentation, people used to, this was the gold standard. Even I would say you should use gold standard of segmentation. If you go back and look at the paper for the dermatology, it really challenges how we segment data. There are 3 papers that have come around this that show even cardiac segmentation. There's no way that a radiologist is looking at the image and saying, this is male or female. I'm not going to be accurate. But the model can learn just the segmentation and that this is male, female, and this is black or white. And so I can send you those papers. But so what we thought was our gold standard, what to me was very surprising, the type of hardware that we use and the location of the segmentation. So if you think about the ventricles and hydrocephalus, which just means the big ventricles for the people who don't work in brain, the way you segment those two and where they are located, if you are not very careful even with your data augmentation, which can also cause problems in medical imaging, then the model can really learn the normal ones are at this level, the abnormal ones at this other level. And so that's going to require a lot more work and rethinking of the way we are doing. Your second comment about CNNs, I think as we've moved to the transformer networks, where you can see the attention is all you need, right? Pay attention to this, pay attention to this other thing. We are seeing much better, sort of don't look all over the place. But the challenge is, how do we deploy these models in the real world setting, right? If your end goal is not just to come up with a new architecture, but you want to deploy the model to impact patient lives, those models are not that robust when it comes to the real world setting because of the processes of like pre-processing, like the brain, the skull stripping, the location. It's too exact and too messy when it comes to actual real world performance.
Hello, can you hear?
Yeah, one final question, and then maybe we should move on.
Okay. Are you going or am I going? Okay, good. Thank you very much for the wonderful talk. I have a comment and a question. My comment is, are we trying to make this model? Well, of course, the goal of a lot of people in this space is trying to make this model to go towards AGI or maybe beyond. But the problem is, if you were to pick an average person and you want them to distinguish between a Ghanaian and a Kenyan, there are stereotypes that they have, right? So these models are just behaving similarly, in my opinion. But again, it's I think one way would be what was suggested by Kyla or from Kyla's presentation yesterday at the ethics meeting or workshop. The idea of thinking it as agentic AI, where then you train model for specific tasks and you try to use different models that try to augment each other's work, improve each other's performance, for instance. My question is that could ties into what you just mentioned a while ago. You know, people build these models and then they add attention where they now identify regions of where the model is looking at. Have you tried looking at that, comparing to what you have done and see if the shortcuts are the same or it's telling you something while it's doing a different shortcut?
So, and this is the abbreviated talk. There's a longer one that I go more into the debiasing. But so the second question is, well, the first comment, I agree with you that these models are, figuring out and cutting our stereotypes. But pneumonia in Ghanaian or Kenyan, I'm pretty sure looks the same. And that's the problem of picking up a stereotype that's not helpful for your prediction, right? And the second thing is, I think there has been work that has looked at where the models are looking. And that's very good when it's disease, right? So for example, where's pneumonia? It should just be in the lungs, something like that. And we can even get new patterns of disease that the radiologist is not very familiar with, but, can be a form of training. The problem comes when I say, look, I can tell that you're Kenyan or Ghanaian with 99, AUC of 0.98. What makes that, right? So these hidden shortcuts are more difficult to capture in the diseases. And just because you don't provide them, just because I don't say you're Ghanaian or Kenyan, then it doesn't mean that the model is not going to use them. It's going to use them because whatever stereotype is encoded in the image is going to be captured. And so that's the sort of like the conundrum of the challenge. Ultimately, I would say training medical data, not just medical images, is very different from cats and dogs. That's what people in Silicon Valley don't understand. And that's where hopefully DSI Africa and the next generation of data scientists is going to help move the needle. And this is because if you train a model to say, is this a dog? If it may never have seen that breed of dog, but it says, this is a dog. It may get the breed wrong. But when it comes to medical imaging, I think we should really say what patterns and then come back and combine those patterns. But that's not how we train the models today. Okay, great, thank you.
Okay, thank you, Judy. Our next speaker is Naja Lau, and Naja obtained her undergraduate and master's degree in human genetics at the University of Pretoria in South Africa. And then she went to University of Witwatersrand, where she's currently a researcher for the Deciphering Developmental Disorders in Africa, or the DDD Africa project. And she's currently completing her PhD in human genetics with a project that's focusing on identifying copy number variants in South African patients with developmental disorders. So the title of her talk is CNV Detection, from exome sequencing data outcomes from the DDD Africa cohort. Great, go ahead.
Thank you very much. Good afternoon, everyone, and thank you for the opportunity to present some of our work that we are completing on the DDD Africa project. So developmental disorders, or DDs in short, have a mental and physical impairment, and even though they are individually rare, they do affect up to 6% globally. And they are very notorious to be heterogeneous. So if you have a look at the picture on your right, you can see that there's quite a complex interaction between the clinical phenotype at the top and the genes at the bottom. So this makes them really difficult to diagnose. And only around 50% of patients are being diagnosed with DD. So this leads to what we call the so-called diagnostic odyssey, where patients are in the healthcare system for many years, seeing at least 7 to 8 different physicians and undergoing a cascade of testing before receiving an accurate diagnosis, or many of them not receiving a diagnosis at all. So just looking at the variants involved in the etiology of DD, it's predicted that around 60% of variants are de novo, so they're not inherited in these patients, and about 40% are single nucleotide variants, or SNVs, and 20% of them are copy number variants, or CNVs. So these are large regions of the genome spanning at least 50 base pairs in size that have been duplicated or deleted. And thus it's quite important to also have a look at your CNVs when trying to diagnose a cohort with DD. So studies like Deciphering Developmental Disorders in the UK, or the DDD UK study in short, has recruited over 13,500 families, and they made a diagnosis in around 41% of their patients by applying exome sequencing and microarray. And thus the DDD Africa study has also followed their model, and we have completed phenotyping and exome sequencing for over 500 families, so trios where possible, child with their parents. And this was in order to describe and define the causes of DD within Africa, and also hopefully to improve the scope and delivery of genetic services. So when we look at exome sequencing, we are only sequencing the regions, the protein coding regions, which represent only around 2% of the genome. Although it has been reported that over 85% of the disease-causing variants are actually within these regions. And thus exome sequencing is quite a good technology to use, and also it can detect SNVs and CNVs with one technology. This is not routinely implemented at the moment, the biggest reason being that there isn't a gold standard for CNV analysis on exome sequencing data at the moment. So when we look at how do these tools actually detect the CNVs from the exome sequencing data, they use the read depth method, so looking at the number of reads within the sequence. And if you look at the top sample number A, you can see that's representing a normal sample. And when you look at sample B in that region, there's more reads than usual, indicating a duplication in that region. And then the same for sample C. When you look at the region there with less reads, that indicates a deletion in that region. When we look at some of the publications that have used this combined SNV and CNV analysis approach within low middle income countries, these 7 studies, an average yield, increased yield, if you look at the blue, that's the increased yield from applying CNV analysis on the data, and an average of around 10.7% in increased diagnostic yield has been found in these studies. However, some of them have increased the yield by over 18%. So for our participants, we've recruited 541 children with DD and their parents where possible from South Africa and the Democratic Republic of Congo. And some of these patients, specifically in South Africa, have also received pre-screening prior to recruitment. So they have been included in these four inclusion categories, and they can meet one or more of these categories, although some of these were mutually exclusive. So in terms of the methods for our CNVs, it was basically completed in 3 phases. So for the first phase, we used an exome data set that we downloaded from the EGA database with known CNVs. So this was basically our truth set that we used. And then we applied the 3 CNV calling tools in order to compare the results of these 3 tools. And then we could move on to our phase 2, was to evaluate the functional equivalence of these tools by calculating sensitivity and specificity. And then we could identify the best tool or perhaps combination of tools to use in order to move on to our final phase, phase 3, where we implemented this on our DDD Africa data set, and then of course CNV classification on the identified variants. So when looking at the results, all 3 tools did have really good results when looking at the sensitivity and specificity. However, Canu's tool did present with the best results. And then at the end, we decided to pair this with the XHMM tool that presented with the second best results out of the 3. And another reason for this was also because the Canu's tool does not call CNVs on 6 chromosomes, and thus using this ensemble type approach, we hoped that this could increase our yield. So for our CNVs that we've identified, this is not a complete data set yet. We are still busy with analysis at the moment, but we've identified 31 CNVs that are likely pathogenic or pathogenic in our cohort so far, 6 of them being in the red duplications and the remainder deletions. And you can see as well the average size of these is around 5.4 megabases, so quite large. And 7 of these have also been validated by using chromosome microarray. When we look at some of the CNVs spanning regions that overlap quite well-known genes and that also are linked to well-known DD disorders, we have identified some of these. And this just reiterates again the fact that even though a lot of the clinicians do pick up these disorders just by looking at the clinical phenotype of these patients, these were missed. And this just again shows the heterogeneity of DD and also the importance of using a genotype first approach and also exome sequencing in this setting. So then in conclusion, we have identified or increased our diagnostic yield by around 6% by incorporating CNV analysis, and this is on our existing data set. So it didn't involve any additional sequencing costs, which feels like a feasible approach for us in Africa and other low and middle income countries as well. This leads to better care and management for the families, and of course can also help to end the diagnostic odyssey for many of these families. So just looking at some of the future prospects, one of our PhD students that are also working on this project and are actually busy with incorporating these tools into an automated CNV pipeline for us. And then another student on the DSI project is also working on the epi signatures, looking at the methylation signatures within our cohort. And then of course, there's lots more to do with the data. But just to mention a few, some of our variants of unknown significance that we've identified, we definitely need to reanalyze and reclassify this systematically, which is a future project that needs to be done. And of course, lots of exciting new development and tools that we can use on our data and also compare to our existing approach. And then long-read sequencing, not only to validate these CNVs and also detect the exact breakpoints, but also hopefully we can implement these on some of our samples that we don't have a diagnosis yet for in our cohort. So I'd just like to thank all of our funders, our partners, and everyone involved in the project and making this possible for us. Thank you.
Okay, well, we have time for just one or two questions. Anybody?
No.
Do you know how the rate of detection for your CNV, like how it compares with some other studies? Or have you done any sorts of comparisons like that?
Yes, for the exams data specifically, yes, so it is around, most of the studies that I found is around, I would say, the 10% mark that they can increase their yield. And as I said, these studies that I've identified specifically also using this in the low middle income countries, the average was also around 10.7%. So ours is around 6%, but I do think that if specifically looking at cohorts where we didn't have pre-testing perhaps, your yield, you're looking at a much higher yield as well, because of course we did kind of filter out those, you know, the obvious diagnoses beforehand by doing some pre-testing. So I think in our cohort specifically, we didn't expect to see like an 18% or a 20%. But I mean, it's still worth it because, yeah, there's other technologies that didn't pick this up. And as I said, we could implement this without additional sequencing costs. So, yeah.
Okay, thank you. Okay, so we'll move on. The next speaker is Dr. Kofi Omega, and he is an associate professor of environmental and nutritional epidemiology at the University of Cape Coast, Ghana, with a strong research portfolio in nutrition and environmental health. And he leads the Ghana Urban Air Quality Project to bridge air quality data in Ghana, and also anchors the Breath Air Project that's part of the Breath Cities initiative. His research focuses on an interface of nutritional and air pollution exposure effects on maternal, perinatal, and cardiovascular adverse health outcomes, using a variety of robust epidemiological and statistical methods, and also leveraging emerging data science techniques. He is a renowned expert in air pollution and health in Africa. His views have been sought both local and internationally in quite a different variety of media outlets. And the title of his talk is Modeling Spatial Variability in the Impact of Ambient PM 2.5 Exposure on Children's Health in Sub-Saharan Africa. Welcome.
Thank you, Kim, for such a kind introduction. And a very good afternoon to everyone and greetings from Cape Coast, Ghana. So we have one of the UO1 projects, which we have labeled DICE. And what DICE means is that data science approaches to improve children's environmental health in Africa. We have 3 aims on that project, and this is one of the aims of the project. So I'm going to share with you some preliminary findings that we have made on this aim of the project. But before I do that, by way of background and justification, the highest number of deaths per capita attributable to environmental exposures, and this is the whole spectrum of environmental exposures, including air pollution, sanitation, etc., have been observed in our region, sub-Saharan Africa. But what is worrying is that the highest burden, disease burden, is always noted among children. And we also know from literature that environmental exposures impact health and development of children and can start as early as in utero and persist throughout childhood and adolescence into adulthood. According to estimates by WHO in 2012, which is quite old, 1.7 million deaths in children under 5 were attributable to environmental exposures and included 570 deaths from respiratory infections. We also a continent that have been documented to have the highest excess mortality from ambient air pollution exposure among children under 5. However, there is very limited evidence from African countries on the early life exposure and child respiratory illnesses, as well as anthropometric deficit. To date, you only have 4 studies that have looked at this relationship. But there were some problems with these studies. So what they did was that they only estimated PM 2.5 from satellite imagery data without integration of data from other sources. And that is something, it's a novelty of our DICE project where we are trying to integrate data from ground monitoring, mobile monitoring, etc. Also, it's important to note that the effect modifying role of neighborhood greenness and then also nutrition in the relationship, as well as how area and household level factors explain the spatial variability in this relationship, have not been elucidated. And that is also something that we are trying to do on the project. The absence of such evidence greatly limits our understanding of the disease burden on the continent and the appropriate policy responses. And I think that is what the DSI project seeks to do. So we're hoping to fill the evidence gap by trying to harness the wealth of exposure data that has become possible by virtue of technological advances with Internet of Things and also machine learning for environmental monitoring. There is also tons of outcomes data as a result of the DHI studies, which is conducted every 5 years in countries. And this data is sitting there and is not being exploited fully. And so we are also leveraging that data on this project. So these are the 3 legs of our project. The first one, which I'm going to share some preliminary findings, seeks to establish spatial variability in the impact of ambient PM 2.5 exposure on children's health in sub-Saharan Africa, and proceed to explore the effect modifying role of neighborhood greenness and nutrition in the relationship. And we have partners in Uganda, in Oregon State, and then also in Canada. And then, so the first aim of the project is situated in Ghana. Then the second aim is to estimate ambient PM 2.5 exposures at multi-temporal scales by integrating land use regression models, high resolution ground monitoring data, and mobile monitoring data in Uganda and Ghana. And this aim is domiciled in Makerere University. Then the third aim is to identify the areal and household level factors that explain the spatial variability in the ambient PM 2.5 child health relationship and establish the temporal changes in this exposure risk profile. And this is being looked at Oregon State University. So for the first aim, what is the hypothesis that we are testing here? The first one is exposure response relationship between PM 2.5, exposure and child health outcomes vary significantly within and between sub-Saharan countries. And then the second hypothesis is that neighborhood greenness and nutrition will moderate the observed relationship. Methods. So for the exposure data, we are making use of satellite-derived PM 2.5 data, which we have gathered from the atmospheric composition analysis group. For the outcomes data, we have taken custody of the several waves of DHS data on a number of child health outcomes. So child nutrition, wasting, stunting, underweight, and anemia, acute respiratory infections. We are also interested in neonatal and infant death. And we have assembled this data from DHS data set that has been undertaken in a number of sub-Saharan African countries. Then we are also interested in the variables that moderate the relationship. So we have been able to obtain NDVI indices from Lancet 7 satellite. We have also assembled nutrition data, and here I mean infant and young child feeding practices data. That is minimum dietary diversity, minimum meal frequency, and then minimum adequate diet has also been assembled from the DHS data set. The data was collected for a period of 1998 and 2022. Countries with no available DHS data were eliminated from the study. Also, countries whose DHS data do not match with the GPS data file were also excluded. So 36 countries satisfied inclusion for the study. The PM2.5 data and then the NDVI were extracted at a 1 kilometer by 1 kilometer spatial resolution. And we leverage data science tools to integrate the exposure data and the outcome data for the epidemiological analysis. So how did we assess exposures among the children? So we did a special linkage of the PM2.5 exposure with each child. So we had, in the DHS data set, you had urban sites and rural sites. So for urban sites, we were interested in 2 kilometer buffer around the household. Whereas for rural side, 5 kilometer buffer around the household. So that is what you see there. Then the J code of each child in the DHS data set was matched with a corresponding PM2.5 concentration and also the neighborhood greenness data. What the matching process does is that it links each child data to an estimated PM2.5 exposure based on their location. And I'll explain how we managed to do that, the prenatal and postnatal exposures. So if, for example, we have a child that was born in 1999, and the annual average, and this child was born, let's say, in September. So it means conception is 9 months. So if the annual average was, let's say, 35 microgram per meter cube, what we do is that we assign that 35 microgram per meter cube to this child, born in 1999 in September. But let's say this child was born, was conceived in September and was born in June of 2000. So 3 divided by 9 times 35 for that estimate in 1999 plus 6 divided by 9 times, if the estimate in 2000 was, let's say, 42, then we add it up. And that is how we managed to assign the prenatal exposures. And there's literature that supports what we are doing. For the postnatal exposure, 12 months. So if the average PM2.5 is 35 for the year, that is what we assign to the child. So what analytical strategies are we deploying on this project? So we are using Bayesian geospatial model to examine the spatial varying effects of PM2.5 exposure on child health outcomes. And this model allows us to control for individual and area level confounders, which we have managed to assemble from the same DHS data. Our geostatistical model will be implemented under the INLA approach, our INLA approach with stochastic partial differential equation strategies. So in brief, this is what we are doing. So you have your data points across the study region. You have a mesh, which is a construction of the spatial correlation. And that enables us to visualize the spatial variability in the impact of PM 2.5 on child health outcomes. So this is our working team. So we have a scientific advisory committee which consists of some faculty from my university and another set of faculty from Makerere, and 2 of them are here in this room. Then we have AIM2, we have AIM1, which is based in Ghana, University of Ghana and UCC, where I'm domiciled. You have AIM2, which sits in my university, and then you have AIM2, which sits in Makerere. We have some PhD students which are interfacing with the numerous MPIs on the project, and then we also have supporting team. So not much to share by way of results because we are still developing and testing our model. We have about close to 1 million children in the study, and this is the proportion of males and females, proportion of children in urban areas, rural areas, proportion of stunted, wasted, underweight children, anemic children, those that had ARI, neonatal deaths, and then also infant deaths. Then what I depict with this is try to indicate proportion of children who fall below the interim target, the new WHO interim target for PM 2.5. So you realize that just about 6.5 children, prenatal PM 2.5, meet the air quality guidelines that have been set by WHO at 15 microgram per meter cube, and much less for postnatal PM 2.5 exposure. And then also we separate the NDVI indices into low, moderate, and high. So these are the proportion of children that you see at each category of neighborhood greenness exposure. So again, children that are exposed, prenatal exposures and high postnatal exposures, you realize it's very, very small amount of children that are exposed to high levels of greenness. So thank you. That is all I want to share with you, and I'm open to any questions.
Yeah, thanks, Kofi, for the talk. Nice talk. So, and also nice data that will be generated. Yeah, that was nice. And also that will come out a great data out of that. So, do you think, as just an hypothesis, that those exposures from mother and infant or child can have a kind of epigenetic modifiers or microbial interactions? And also, are you looking all those aspects? That's the first question. The second question is about the model. So you state that you'll be applying spatial model. So are you thinking from the literature perspective, and given your data is a demographic data, understand there will be kind of a high level of heterogeneity in the data? I don't think partial model can actually lead you to an accurate result. Take into account that your data will be really spread across, the country or the regions. So are you looking as an alternative model like Bayesian Dirichlet processes to model efficiently your data? Thanks.
Yeah, thank you, my good friend Emil, for the question. The first part, yes, that is certainly what we are interested. We are also interested in genomics, but unfortunately, we don't have genomics data in the DHS data set, and we don't have any way of getting such data. So that is not something we can test. But of course, there are a number of studies that have looked at gene-environment interactions on child health outcomes. And that is something that we are pursuing in my group. And in recent time, we have set up a BEV cohort in Cape Coast. And we want to leverage that BEV cohort to collect these genomics data and do some of the wonderful things that you have proposed. On your second question, in fact, as I indicated, we are still developing and testing our models. And Justice, who is our biostatistician on the team, is in the room. So I'm sure he has taken note of some of the shortcomings that you have indicated. And definitely we'll toss you over during the dinner at the beachfront. and then possibly I'll come back to you on that. Thank you very much. Oh, Justice, you want to, yeah. Justice is a PI on the project, and I'll give him the opportunity to, yeah.
Yeah, thank you for that excellent question. So we don't foresee a challenge. The reason being that we are using the cluster locations as a unit of analysis. So during our analysis, we will now extrapolate beyond the sample locations to cover even areas where data is not coming from, which we call kriging or interpolation. And we have mechanisms in place like the mesh, the SPD strategy that has been doing this for some time now. And in our previous work, like Prof actually showcased, that is a similar process we followed to develop the map that Prof just showed to you. So when it comes to the uncertainties and everything, we do quantify all that and get the back into the model. So when it comes to that location, I can assure you we don't have too much noise around that area. But if it is other outcomes, yes, we can discuss that. Thank you.
Okay, you had a question.
Yeah, let me follow up a methodological question. I'm Charles Michelo from Across Research in Zambia.
Please, can you speak up a little bit? I can't.
I'm Charles Michelo from Across in Zambia. You have used DHS data pool, and I do note that, of course, you have used cluster grouping, and some people have argued that even when you do so, because of the different sizes, you must first manipulate your data using small area methodology, which you haven't alluded to. Do you think that interferes with your estimation? Thank you.
That's a big one. I think I'll allow Justice to come in again here. Yeah.
Okay, so the first, when you started, I was even thinking you're going to talk about the displacement that happens in the DHS to avoid some kind of identifying individuals, but rather went on the other side, which is not too much a problem in our work. So what we're doing here is that the locations where the children are sampled from, that's what we call a cluster. And in each cluster, you might have maybe 10 children sampled under 5. And out of that 10, you might have, let's say, 3 of them coming out as stunted or underweight, or let's say wasted or had an anemia. So all that we are doing is that we estimate the prevalence at the cluster location. Assuming there are 10 and 3 had anemia, it will be 3 over the 10. So what we do is that given that prevalence, that cluster location, which is indexed spatially using a longitudinal latitude, we now bring in exposure variables. And then what we try to do, we don't aggregate at a district or region at the start. We model at that individual cluster location, taking into account the displacement. After that, we then map across the entire country. Then we can use the regional or the district polygons of the country to actually estimate the prevalence if you want to do it at the district or the regional level. So that actually takes into account your fears already because we didn't aggregate. Thank you.
Okay, I think in the interest of time, we're going to take one last question from her and then move on.
Hi, I'm Megan Fitzpatrick from the University of Maryland at Baltimore. I think it's really interesting how you're doing the prenatal and the postnatal. And for the postnatal, you said you're following them for 59 months, right? Do you think that it's possible that some of the reason for the increase in the air pollution for the older children is because it is such a long period of time and that air quality may be degrading over that period? And is that a hypothesis that you're planning to explore within this work and think about what that might mean for the future? Or do you have another explanation for the difference between the groups?
I'm not entirely sure what you mean by the question, but so you have, in each country, you have a number of children. And then these children are under 5, you know, born in different months, in different years. And some are in the study for throughout the 5-year. So you have the DHS study is conducted at a time point, and you're likely to encounter some of these children in the next iteration, you know. So the estimate that you get, the PM 2.5 that is assigned to that child, is assigned at one time point. It's not cumulative. Yeah.
Oh, okay, so it's, so the number that you're assigning to that child for the postnatal
Yeah.
fraction is not a 5-year, is not,
But the children are in there with different ages. So you
Okay,
might find a
got it.
child 2 years, 3 years, 4 years, 5 years.
okay, then
Yeah.
please disregard my question. I misunderstood.
No, that's
Thanks.
fine. That's fine.
Okay, just one very quick question. Thank you very much.
Okay,
I
thank
was
you.
just
ma'am.
wondering whether you took into account indoor pollution and whether you collected, apart from satellite model data, whether you also collected data. How granular are those, if you did, and what did you use to collect? Thank you. Mayowa Wulabi.
I mean, you mean the air pollution data?
Yes.
It's very granular. I mean, satellite data is very granular compared to even ground monitoring. But of course, there are limitations because the overpass is just 1 hour and it's measuring the whole entire atmospheric column compared to what the ground monitoring data. But for this work, we are not necessarily integrating ground monitoring data because we don't have ground monitoring data for all the sub-Saharan African countries. But what we are doing on Objective 2 of the project is to develop land use regression models, and then we'll test its applicability in the different African settings and also exchangeability between different cities in Africa, and how that can, for example, not necessarily replace the satellite imagery data, but how that can also improve exposure assessment. Because one of the things we want to also do is to how to address the exposure assessment limitations, which bedevils environmental epidemiological studies.
Yeah, so what did you use to collect the ground monitoring data? And then did you account for indoor air pollution?
No.
Beyond what the satellite collects? Yeah.
For this, the DHS data has proxies for household air pollution exposure. So the household air pollution exposure for the uninitiated is pollution as a result of using biomass and other dirty fuels for cooking. But you know, there are huge limitations with this because it's a dichotomy. Are you using a biomass fuel? Are you using clean fuel? And also the question is on your primary cooking fuels. There's also fuel stacking in most of the household. So we don't think it's prudent to control, to bring that into the analysis. But of course, that is something that we can consider based on, I mean, comments like yourself. But it's not something we have thought about in terms of trying to adjust for primary cooking fuels in the models. But again, we have a number of socio-demographic factors which we are adjusting for in the models. And because of multicollinearity, we might have to be careful in throwing all these covariates into the models, because some of these socio-demographic factors, I can bet you, can correlate with type of fuel that is being used in the household. Okay. Okay, thank you very much.
Yeah, and so our next speaker is Dr. Rahil Bekene. Thank you. Dr. Bekene is from the School of Information Science and Addis Ababa University. Her research interest is using data science techniques to improve health outcomes for children and mother. She's a principal contact from Ethiopia for the Africa DST project. In today's talk, she will talk about lessons learned in public health from Eastern Africa through data science training. Thank you.
Thank you for the introduction. Good afternoon, everyone. I'm sure I will not make you sleep because I will be talking only for 2 hours. So the topic is impactful research in public health data science, the lessons learned from AfriaDST project. So I'm not going to present research results, rather how we do the research. So let me give you a brief, quick overview of how I will be covering the presentation. I will start by highlighting the public health challenges in Africa. I'm sure many of us are aware of the challenges in Africa, but I would like to briefly revisit so that we give context to the AfriaDST project. And then I'll say a little bit about the role of data science in addressing public health, again, to give highlight for the AfriaDST project. And then this will be followed by activities, lessons learned, and way forward. So the AfriaDST project has been presented in a number of times, both online and also in this conference. So it's a collaboration project of 3 universities in 3 countries: Columbia University in USA, Addis Ababa University in Ethiopia, and University of Nairobi in Kenya. So all of the PIs are here. So I'm practically with my bosses. So again, under each university, it is kind of interdisciplinary. We have academicians from public health, from School of Information Science, where I come from, and also Department of Statistics collaborating. So as you see, all in Columbia and University of Nairobi, we have public health, statistics, and technology background academicians collaborating in the project. So coming back to the public health challenges, as I have said, we will revisit even the ones the speakers prior to me have said. There are high maternity and mortality rates, high prevalence of communicable and non-communicable diseases, limited health infrastructure. Why we are brought here together is probably to address the inadequate access to timely health data, and also an ability to make informed decisions based on the data. And one important thing that I would like to stress is health workforce shortage in the area of data analytics. So considering this, we can say a lot of challenges in Eastern Africa. One of it is the siloed efforts, that is collaboration gaps between the academia and industry. Normally, it lacks stakeholder engagement. In most cases, stakeholders are excluded from various projects done by the academicians. So what is the role of data science in addressing public health challenges? Basically, we want to address or to discover the hidden patterns within the data. There are a lot of interventions by the government, but we don't need as such an improved public health in many areas that need to be tackled with. So we want to harness the vast amounts of health data and would like to make a transformative solutions to tackle the complex public health challenges. And as such, we need to collaborate. So in terms of the public health data science gap, we see severe shortage of skilled data scientists. As of even now, as I'm speaking, we teach our students by getting mentorship from Columbia University and other institutions who have expertise. So we have limited human capacity, and there is significant lack of trained data scientists in the health sector. And we also have challenges in data access and quality. What we have discovered actually when we started the AfriaDST program is every stakeholder says they have data, but looking deep inside, they have aggregated data rather than having lots of attributes that would like to consider the relationship and get the hidden attributes. So as such, there is also lack of data, although we say there is vast amounts of data. So would like to prepare our students to also prepare the data that can be addressed by data science specialists. specialists. And the other is data quality issues. And what we have considered as one of the problems is also data privacy and security, especially from the viewpoint of stakeholders. So there is limited research on local health issues. So there is underutilization of existing data. Even the limited amount of data is not properly addressed. So for this, we have initiated the AFRIA DST project. So one of the project objectives is the MSc program in Public Health Data Science, which we have established both at the University of Nairobi and Addis Ababa University. We have faculty development program so that we strengthen the capacity of those scientists teaching in the program. We also have short-term training programs for a wide spectrum of trainees. But one of the innovative things that we have done is research immersion. The students do not just do their research, but they join research groups of various domain areas, and they do their internship and actually work on real-world problems. So regarding the MSc, Kenya graduated its first batch in September 2024, and it's going for its third batch. It has a total of 50 students currently ongoing in the first and second year, in addition to those who have graduated. In fact, one of them who have graduated is an early career researcher who presented her work in the morning. And in Addis Ababa University, we have two cohorts doing first year and second year. So the early career researcher who came from Ethiopia is also in his second year. He presented his poster yesterday. So the focus is more on research immersion and practical training, which we call impactful research. So we do institutional collaboration. We collaborate with various academicians in various disciplines. So we call it interdisciplinary approach for our MSc programs. We have signed various data sharing agreements, even though, again, the data that we have discovered is very limited. And the students do internship. So one of the things that we also do is mentorship and joint supervision. Our faculty development program has been very successful so far because we get different kinds of mentorship from Columbia University, and our faculty scholars also teach in the program. And students receive various perspectives on the research methodologies, ethics, and practical applications. This has actually helped refine their research focus. So we actually want our students to get real-world data, and we try to improve their problem-solving skill. And that's how we are trying to make our address the impactful research in the works. So the case example is in AFRIA DST research working groups. In Ethiopia, we are more or less grouped in domain areas. We have data science for maternal and child health, data science for environmental health, and we have communicable and non-communicable. And because of the fragmented data and unavailability of data, I would like to create some kind of integration. So we have established a data warehouse architecture research group. And in Kenya, there is big data analytics, data security, privacy and ethics, and genomics as a research group. So we help our students get a refined research focus because members of the research group come from various areas. And as much as possible, we are trying to ensure high quality outcomes from the research that the student take by shaping the direction of their research. We have also student-student collaboration because we have student-led seminars, and the students also cooperate with each other in finding problems and trying to solve the problems on their own. And this is all to make them be aligned to the public health goals and real-world research immersion. So, so far, we have published 3 works, but some of the impactful research application we have done are One is the spatial temporal analysis of maternal mortality determinants, which is yet undergoing review. We have image classification of TB, pneumonia, and COVID-19. So maybe I can answer Judy's question here. We have some kind of image classification research. Air pollution prediction models, which is actually published by Valin, who has presented her work in the morning. We have also some ongoing research on economic burden analysis of respiratory diseases and brain tumor detection using MRI and deep learning. So these are all projects of students and the faculty scholars working together. So you'll find 2 of our publications online, and we just got accepted in Frontiers in Public Health on one of our research articles. So these are the things that we're done. I would like to wrap up by giving you some information on the lessons learned. We have learned that the importance of collaboration, collaborative partnerships, especially in terms of bringing people from various fields of studies together, so that the expertise of one will contribute to the ongoing health research. So interdisciplinary research and ethical collaboration. We have ongoing challenges in managing balance between data access and privacy concerns. So especially in terms of getting stakeholders giving us the data, either because they don't have it or there is a data sharing policy that's going on. So the other thing is the way forward. Would like to scale up our capacity building initiatives. So apart from the MSc, would like to go ahead and create other real life short-term training programs which addresses the public health data science needs, and probably open PhD programs in the future. So we would like to enhance our collaboration both within the projects of DSI and outside. So a peer-to-peer mentoring system is one of the things that we'd like to do. And basically, we want to focus on practical hands-on data science tools, because at the end of the day, this is what we are missing as a shortage of workforces. So that's how we plan to build sustainability of the project at hand. So thank you for your attention. If there is any question.
Thank you. We have time for a few questions. Okay.
Yeah. Charles Mitchell again from Across. One of the things that you mentioned is the challenges regarding data availability. And in many cases, what was said, there's even no data, even the little that's available, literally not available. But I would like to pick up that point because one of the major problems we do see in Africa is even when data is available, some of the inability determinants are the fact that there are no systems. Sometimes you don't even know who to write to, who to ask to access the data in country. HIV, tons and tons of data that's available, and yet people struggle to access it. Now, your program relies on availability of data for it to flourish. How have you addressed the issues of data access, privacy on the research domain? Thank you.
Yeah, thank you for the question. In terms of data availability, we've tried to sign data sharing agreements with various institutions. That's how we have brought them on board, actually. We have a data sharing agreement with Ministry of Health, the Ethiopian Public Health Institute, in fact, which has a lot of data. And we have the Central Statistical Services. And also there are others like Meteorology Institute, Ethiopian Agricultural Institute. We have tried to sign the data sharing agreement. We have actually signed with some of them, but the issue is when we send our students to for internship, the students do not get actually the data that they are looking for. Either it is an aggregated data, so they don't have the required attributes that they would like to work on. So we are trying to address these issues by going into the data which are actually existing on paper, because the data are on paper. That is the problem that we have currently in Ethiopia. The EPHI, which is a major host of most of the data sources on health, is on the process of approving its data sharing guidelines, data sharing policy, which is actually taking longer than expected. But our students are trying to get data as much as possible, so we encourage them to get a real world data. Because at the end of the day, what makes impactful work is for them to get some kind of data, even if very small, to have a real world data. Otherwise, I share your concern that the data availability is problematic. Thank you.
I was really interested to hear you talk about the data warehouse architecture working group. And I think it also speaks to the question you were just discussing, which is the variety of formats or incomplete formatting that the data come in. Could you tell me a little bit more, or tell us a little bit more about how that works? And also, in particular, are you creating templates for clinicians or other public health practitioners to use as they begin to collect their data? Or are you creating templates for your trainees and your data scientists to use as they collect the data from all of the various sources and put it into your repository, or both? Thanks.
Thank you. Our research groups just started with new students who are working on their internship. Unfortunately, no one joined the data warehouse research group. So they are into communicable and maternal and child, where there is data. But we're trying to make them come, because right now the research group is composed of instructors from public health, Ministry of Health, and IT fields of study, but would like to take more options, opinions on how to best handle these things, because we find that there are scattered data fragmented everywhere. So we'd like to bring them into together. So I would be happy to discuss this with you further. Thank you.
Thank you. We'll move to the next speaker. Thank you.
Thank you.
Our next speaker is Miss Ruth Najane. Who's the speaker? Oh, two of you? Okay, so I only got one name. Okay, so Miss Ruth Najane is a Kennedy Trust Prize Scholarship Scholar. Her research interested is on computational analysis of the human nucleoside antigen. She's currently a PhD student in cellular and molecular medicine and Kennedy Institute of Rheumatology. So we have the second speaker.
Oh, that's not the PowerPoint.
Who's not? Okay. It looks like there's some technical issue. We will move to the next speaker. The next speaker, there's a little bit change from the agenda. Our next speaker is Dr. Catherine Juniat. Dr. Catherine Juniat, she's a professor from UCLA. She's also a surgeon. Her research has been focused on public health and health equity. She tries to identify risk factors driving disparities in surgically related disease and treatments and disparity in treatments. So she aims to find innovative ways to overcome those disparities. So in today's talk, she's going to introduce a toolkit on how to estimate the different wealth in different countries in Africa. Let's welcome Dr. Julia.
I'm struggling to use the pointer. Oh, there we go. Thanks. Okay, so I apologize. My colleague, Prof. Ngwefak, was going to speak today, but he couldn't make it, so I'm filling in for him. And I'm going to talk a little bit about our hub, which is called Design Africa, and then speak about one of our projects on health equity surveillance. I'm a trauma surgeon, as you heard, so I'm going to focus more on the framing and rationale for this problem and the results and their application rather than go deep into the methods. The goals of our hub overall are to decrease the burden of injuries and surgical diseases in sub-Saharan Africa and improve access to quality surgical care in Cameroon and other similar countries. The main three aims of our hub are really doing this through research, networking, and capacity building. Our hub is structured like probably many of yours. In addition to the admin and DMAC, we have a capacity building core. It's led by Professor Shishom and housed at the University of Buea, where he's faculty, and our executive director is Dr. Disak Delon, who's here today with us. We have several projects and seed grants and pilot projects as well. And I'm going to focus today on this one. This is the Health Equity Surveillance Project. So just to back up a little bit, injuries are a huge problem. I'm a trauma surgeon. I care a lot about injuries. And unfortunately, they kill more people annually than HIV, TB, and malaria combined worldwide. About 6 million deaths annually are estimated. And although a lot of people tend to think that injury is a high-income country problem, this disproportionately impacts LMICs, with 90% of the injury-related mortality occurring in LMICs. Road traffic injuries, or RTIs alone, which are only a proportion of the injuries that are caused worldwide, is estimated to become the 5th leading cause of death by 2030. And what's not often talked about, in my opinion, is that in addition to the people who die, that's just the tip of the iceberg, because injury tends to disproportionately impact the young, healthy members of society who tend to be primary caretakers for their household or primary breadwinners. So when they get disabled or have other economic outcomes, including economic consequences of injury, it has a pretty impactful result on a societal level. Okay, so let me back up one before I show that. But what we started doing to just first address this problem was collect data. There were not a lot of data available in sub-Saharan Africa, and very few available in Cameroon. So we started by creating a trauma registry, which means at the Central Hospital of Yaoundé, we implemented a pilot trauma registry where we collected data on the injured patients who presented there. And one of the things we wanted to track was the socioeconomic disparities that were potentially occurring. We knew in the United States that injury, people who have risk factors in the social drivers of health are more susceptible to being injured and tend to have poor outcomes. We were curious if this might be happening in Cameroon as well. So we used a method that is used by the DHS. I know we spoke earlier about DHS data, where they take about between 20 and 30 variables. At the time in Cameroon, it was 26 variables. And they take these and use PCA, principal component analysis, to create a wealth index for each patient. We did this in Cameroon at the Central Hospital Yaoundé about 15 years ago, 10 years ago, over 6 months. And this required 24-hour-a-day research assistant presence in the hospital, in the emergency ward. And they asked each injured patient all these questions. And then we calculated the wealth index, and we compared this to the general urban population from the DHS data from the year before in Cameroon. And if you look at this figure, you can see that in the green, they're just the quintiles, which by definition are all equal, of the DHS urban population in Cameroon. We took the cut points for these quintiles and applied them to our data, which are the injured hospital care seeking patients in our trauma registry, to see who is coming to the hospital. And as you can see to the right, the higher or wealthier quintiles were overrepresented in our care-seeking population, injured population. Additionally, when we looked at injury severity score, the poorer wealth quintiles were more severely injured. They had higher injury severity scores. They had a higher threshold for seeking care based on how injured they were. So this led us down a path of looking at trauma and wealth. We did a number of studies. One was a community-based survey led by Dr. Christie, who's also part of our team and is here, another trauma surgeon at UCLA, in partnership with Professor Chichom. Others looked at our Cameroon Trauma Registry data, which we subsequently expanded to 4 hospitals. And we found a lot of patterns. Poorer patients were less likely to seek formal care. They're more likely to be severely injured. One of the main deterrents to seeking formal care was the expense that was associated with it. And when people did seek formal care, they often had financial toxicity afterwards associated with healthcare costs. And people with health insurance, which is the minority in Cameroon, most pay out of pocket for hospital care, were more likely to get admitted, which suggested that even getting proper care was somewhat precluded by people's ability to pay, not surprisingly. So this really led us to become more and more interested in studying the inequity in access to care and health outcomes associated with these issues. So here's the problem. Our status quo is that the poor have worse access to trauma care. And where we want to get is to eliminate these disparities in an ideal world. And the challenge is, to do that, you have to have a way of measuring disparity, creating interventions, implementing those interventions, and then measuring the disparity again to see if your intervention had the desired effect. The problem with the current methods is that these require a lot of time, resources, and just aren't really feasible in an emergency setting. You can imagine if you were in a car crash and came in and were at best in a lot of pain, or at worst in a life-threatening situation, the last thing you or your family want to do is answer 26 questions about what the floor of your house is made of, or what fuel you use to cook, or whether or not you have a cell phone. So this is not a feasible way to do longitudinal wealth surveillance in an acute care setting. So this led to our Project One, which is basically creating a more efficient way using less questions to achieve the same result. We propose to develop economic cluster models for all the countries in Africa that have available recent DHS data, validate these by testing the association of our developed clusters with outcomes, both social and health outcomes that are known to correlate over through the literature over time with wealth, and then implement these in not only our own Cameroon Trauma Registry, which now with this current award has been expanded to 10 hospitals in 7 of the 10 regions in Cameroon, but also in 2 partner sites outside of Cameroon, one in Uganda at Soroti Regional Referral Hospital, and another one in Cape Town with our partners at UCT. Finally, the last aim is to take this toolkit, because we'll have a toolkit for each country with the available DHS data in Africa, and make this publicly available. So researchers in other countries or other disciplines can use these to track wealth in their own study populations. Again, I'm going to be brief about the methods because I'm a trauma surgeon, and the biostatistician who was supposed to be here today presenting is not, so bear with me. But as we took the data from the countries that had relatively recent data, we eliminated variables that had significant missing observations or lacked sufficient between household variation. And we chose composite variables over more complex individual variables. We then created the economic clusters, and this was done through a partitioning around medoids or PAM method, which is a non-parametric centroid-based clustering method that selects K data points or medoids. And then it conducts a heuristic search across all the households to select the medoid, which is the observation closest to all the other similar observations or subjects. And then we made an objective judgment of the clusters using the average silhouette width. The validation, we use 2 outcomes that are used commonly in DHS data. One is child mortality, and the other one is women's highest educational attainment, and these have been shown to correlate with household wealth. And then we added another outcome, which is the access to primary formal health care, which has also been shown to correlate with wealth. And interestingly, this one has been shown to correlate with wealth in conflict zones, which was of interest to us as well because of some political unrest in 2 of Cameroon's 10 regions, and we are present in those regions with our study. And then we basically tested to see if these clusters were associated with these outcomes as one would predict if they were behaving as you would intend them to. So what does all this mean? We're taking, in summary, the DHS Wealth Index approach and changing it. Instead of 20 to 40 variables to get a score of wealth that is then broken into quintiles, we create clusters. We narrowed it down to 4 variables through this method, and that basically bundles like with like. So the patients most similar to each other in terms of their assets get grouped in a similar cluster. Here is the outcome when we looked at our results when we looked at our 3 outcomes. Here's the results for childhood mortality. And we presented this in a kind of forest plot because we wanted it to be familiar, kind of like a meta-analysis, but this is not a meta-analysis. This is the relationship between each cluster as an ordinal variable and the outcome of interest. And here's childhood mortality. And you can see they're all strongly positive except for one country, which is Chad down at the bottom. And this indicates that these are associated with health outcomes and that the wealth clusters are behaving as one would expect, and there's consistency across this in all 3 results. Here you can see the education outcomes. Again, Chad is the outlier, and then the primary healthcare source. Chad and Niger outliers, and Gabon is close, to not being positive, but all the other countries really do behave as indicated. So what does this mean? Well, one more thing. This was something that we didn't go into this intending to have this particular aspect, but we found it more logical, I guess I'll say. If you think of the DHS Wealth Index quintiles, which are on the right, you can see that because they're divided into quintiles, if a population has a group that's larger and more similar, that group gets kind of dissolved over the quintiles instead of being stuck together. So that's an artifact of forcing a quintile formation onto these. Whereas with the clusters, it allows for the actual heterogeneity of the population to be more represented. So we thought that this was another reason that the economic clusters were advantageous. What do we use this for? So now we've implemented the clusters. Cameroon now has 4 questions instead of 26 in our trauma registry. We just implemented these in Uganda and South Africa. The data collection there is underway. But for the Cameroon, we did a recent study. This was led by Dr. Disak, our hub executive director, who is here today in conjunction with Mark Yost, who's one of our surgery residents who spent the year in Cameroon last year working with this group, and looked at protective gear. So protective gear is helmet use when you're riding a motorcycle, or a seatbelt use, or a car seat or a booster seat for a child of the appropriate age. And what we found, if you look here, this cluster 1 on the left is the richest or wealthiest cluster, and cluster 5 on the right is the poorest. And you can see for protective gear users, which is the light blue bar, the richer clusters were more likely to be using protective gear. And when we looked at mortality and major disability as a bundled outcome, they had better outcomes in their trauma care. The poorest clusters had the opposite. So again, this trends the way we would expect it to, unfortunately, and gives us a target now to talk to the Ministry of Public Health about the disparity here and the factors that may be preventing people from the poor clusters to be able to use helmets or other protective gear that are life-saving. Here again, this is the adjusted odds ratio from the multivariable analysis, again showing that the poor clusters have worse outcomes. Where are we at now? We're writing up the manuscript here, and that manuscript will have links to the more detailed treatment of the data, cluster formation, and the code. But ultimately, this year, we're going to be focusing on developing this toolkit. So the outcome will be to have a website where if you're a colon cancer researcher who's doing work in Tanzania and you want to see if there's an issue with access to care in your study population, you can take either the code that you can freely download from our website. You have the 4 questions you need to ask your study subjects to track this, and then you can assign clusters to your population. Or we want to make this as accessible as possible. So we're trying to incorporate a feature where if the coding is too cumbersome, the data can be uploaded and run on our server for you, and then clusters are assigned to your study subjects for you. So we're hoping that this mitigates a barrier to studying health equity and outcomes associated with wealth in Africa. I want to thank all of you for your time, and thank you the moderators, and thank you to our team who is amazing, and I'm so grateful to be part of this group. It's such a wonderful conference. I appreciate it.
Thank you for the interesting talk. Questions?
Thanks for the talk. It was great. Hopefully none of the creators of the Wealth Index are in the room, but it's nice to see, because it's just not representative of the distribution of wealth within each of those clusters. It's so broad, it doesn't represent. Which 4 questions did you land on, and have you found that they're consistently representative across different countries?
No, it's so interestingly in Cameroon, the 4, I'm going to remember them right now: cell phone, TV, mixer, and cable. Yes, no. And then we actually found, interestingly, that most of the countries have a number of different combinations of assets that work relatively equally well. So we plan to publish that as part of, each country will have its profile on the website. And those questions will all be, all the ones that we think perform well enough based on the ASW will be included. And then people can choose based on their context which one's easiest for them to ask. And sometimes there's cultural issues with asking too many questions, or certain topics are sensitive and you don't want to include those. So it gives some flexibility to the investigator.
I have a question. So when the patient is asked for those 4 questions, they will be coded into one of those 5 clusters?
Yeah.
So which cluster they will be placed into? Will this impact which kind of treatment they will receive?
Not in my hospital, hopefully. I mean, ideally, no. I think what we would do, because in real time, the clusters have to be assigned retrospectively. So in real time, it shouldn't influence the research assistant. Currently, we're fortunate we have research assistants kind of in addition to healthcare providers. So it's a little bit separated, but that's a good question, you know.
Yeah, thank you. Let's thank the speaker again. Okay, so let's move to the last presentation of this session. We have, besides Ms. Ruth, we have another speaker along with Ms. Ruth, is Professor Segang Fantuma. Professor Fantuma is from the it's a Queen Mary University. His research expertise on the genomic diversity. He's also the head of the genomics and MRC Uganda. So he's a PI of the BCX Africa project for the DSI Africa. So both of them, they will talk about how to use a polygenetic risk factor to estimate red blood cell traits. So let's.
Thank you for the introduction. And this is the last talk for the day, I think, and you can go for dinner. So it's not time to sleep. We're going to be talking about BCS Africa. This is a project where we're trying to develop genetic risk score to predict a risk of disease, different kind of disease. So this project is led by three of us, myself, Ama, Kuma, who is there in the audience, and also Tinashe Chico Warrior. So the objective of the project is twofold. So we are trying to, first of all, aggregate data all across the previous initiative, that's the Asia Africa. There have been so many large-scale genomic studies that have been done in Asia Africa. So the first objective is to use this data and to do the large GWAS of blood cell trade in about 40,000 individuals across Africa, and also leveraging other data sets on the databases like MVP, other African ancestry in the US, in the UK Biobank, and other places. So the second one is because the African genetic architecture has a lower linkage disequilibrium. So we want to leverage this to refine in order to find potential causal variant. And the third one, which we'll be talking about today, is develop PRS that can help to predict disease risk. And the last one is to either use this PRS and also other risk factor to see whether they are causally associated with other different disease, particularly NCD disease, all NCD disease, particularly focusing on African population. So here on the right in the figure, you can see the overall of how we are trying to aggregate this data set. Okay, Ruth.
Thank you so much, Bob, for the introduction. Hi, everyone. I'm Nanjala Ruth, a PhD student in Oxford, and I'll be speaking about the project that I did with the team, and I'm presenting on behalf of the team as well. So the reason we are interested in working with blood cell traits is because traits such as white blood cells, red blood cells are very important clinical markers for hematological and cardiovascular diseases. And therefore, we just want to try and understand, especially when we say that predicting disease outcome is poorly understood in Africa, and also that the genetic determinants of blood cell traits is also poorly understood. So we want to try and sort of like bridge that gap. Although we know that from GWAS studies, most of the discovery data sets are from the European ancestry, but now with the advent of machine learning algorithms, it has been shown that we could actually use these algorithms to predict in diverse populations. So we want to test this in African population and see whether we can get better predictivity using machine learning algorithms. So what are we doing about this to address the gaps that I've spoken about? So one is being done by IAD, who's working on generating a discovery dataset based on GWAS meta-analysis using both continental and diaspora African datasets. And I'm gonna speak about what I did and what I'm doing, which is developing blood cells traits polygenic risk scores. So what we are doing is using a machine learning algorithm, which is ElasticNet. So this has already been done in diverse populations, and we just want to test how it performs in African population. So ElasticNet sort of like uses penalized regression, which is basically just selecting scores that are applicable to the particular population you've given it, and then working with scores that work for that particular population. So how are we doing this? I have written an Nextflow pipeline, which would ensure reproducibility, reusability, and transparency, which means that we can be able to use this pipeline for different data sets and not just the one that I sort of like worked on, so we could apply it to different African population data sets. So this pipeline is publicly available on GitHub, and you can scan me as it says there. So you can see the kind of sort of like pipeline I put together. So the reason why a pipeline is important is, a keynote speaker spoke about shortcuts. So when you're working with huge data sets, particularly for this case, so you can imagine if you're looking at mean platelet volume, which is what I've highlighted there, and for example, hematological measurement has 400 score files. So this means that you have to go to the PGS catalog and download all the 400 files one at a time, and then compute a PGS scores on each of them one at a time before sort of like combining them into an integrated score. So this is why a pipeline is important, because it's already written. It means you just have to key in the IDs for that particular mean platelet volume, and the pipeline would sort of like run all the analysis from, which is what I'm going to talk about next anyway. So the pipeline, this is what it does. So it downloads the score files from the PGS catalog, and then it modifies the score files in a format that is acceptable for computing PGS scores. And then after we have computed the PGS scores, we build an integrated score using ElasticNet, and then we can now test it or evaluate its predictivity. So this is what the pipeline does in theory. In practical, this is how it looks like. So for example, if you're working with 15 blood cell traits, each of them has different score files within it. It means you just need to key in the IDs for those particular traits, and then the pipeline runs everything until the end, where you end up with your 15 blood cell traits and their integrated score, and you can now do sort of like downstream analysis. So this is what is publicly available, but then I also did put it together in our website, a Quarter website, so that because we are working as a team and we want our collaborators and team members to be able to see what I'm actually doing and that I'm not, to ensure transparency, which is what we talked about. So we put it on a Quarter website, which is what is on the left or right, based on where you are, which shows all the analysis, all the downstream analysis, step by step, and for all the 15 traits that we tested, and for all the PGS scores that we tested. So what I did was I did both the integrated score and the individual based scores. So that's why you see a lot of sort of like data on the left side. But on the right, it shows a snapshot of the results from MPV, which is the mean platelet volume that I spoke about initially. And it shows what the ElasticNet model does and how it's sort of like, that is the results that come you end up with after running ElasticNet model. Right, so as I mentioned, I showed the results for MPV, but then I did this in 15 other blood cell traits, which you can see here. And as I had also talked or mentioned before, I did both the integrated and the individual based analysis, and then just picked the best individual score and sort of like put it or compared it with the integrated one to see whether if you use an integrated score, you get better results than using an individual score. I mean, we expect that the integrated score will perform better, which is what we saw, so that was good. And also the mean platelet volume had the highest score, which is what I showed you initially. So this is just preliminary results. But moving forward, as I had mentioned before, we are working to pull data together to work on the largest GWAS for blood cell traits in Africa, and also looking to evaluate the integrated PRS score. Now that we have a pipeline, we can easily just test other Afrocentric data sets. So like the African data sets within the UK Biobank, and also the Malawian data set. And then after that, we can evaluate its predictivity to a range of non-communicable diseases in Africa. I'd like to acknowledge the entire team in the BCX Africa project, and also to acknowledge the University of Oxford and the Kennedy Trust, who sort of like funded my sort of like exchange program at Harvard Medical School, where I did this analysis with Dr. Tinashe Chikowore, and also to thank everyone those that are both in the audience and those that are not here, but you can see their faces and their names on the slide. Also to just highlight that two of my colleagues have also talked about some of these projects that we are doing as a team. So there's Chisom who spoke this morning, and also Chris who had a poster. So in case you haven't seen Chris's poster and Chisom's poster, this is your sign to go and look at them after this or during the break session. And with that, I'd like to say thank you.
Questions?
Yeah, thank you very much, Ruth. Very wonderful presentation you have here. Congrats to the team. I wanted to ask, your last statements talked about it will help you in the predictability of NCDs given the different blood traits. Where the blood traits taken before the NCDs occurred, or were these two things taken at the same time, and how would you determine temporality in this case? Yeah, thank you.
Thank you. Prof will answer that question.
So what we're trying to do is to use genetic to do the prediction. So it doesn't matter whether it's taken before or after, because your genetic does not change. So if you were here when I think Chizo was presenting our work, so you use a genetic to see the blood cell trait, and you see whether that blood cell trait can predict something. You know, in case of Chisholm, it was diabetics. In case of for Chris, it was a kidney function. And we could use that also to predict anything, to predict any non-communicable disease. So it doesn't matter where, at what point you test people for blood blood count. What matters is that their genetic, which you are using to as a proxy, doesn't change.
Yes, thank you for that good presentation. I'm just following up on the last speaker's question. I heard a term like tempora. For us in the modeling framework, when the term tempora comes in, it's like we are looking at something over time. I think that is where he's coming from. So if the prof can come again, that was not answered. Thank you.
Thank you. If no more question, this will close this session. Thank you for being here. Enjoy the rest of the day. Thank you.
Yeah. Thank you. Thank you for the speakers and all the great questions.
Yeah.
thank you to all the facilitators and the speakers today. Oh, yeah, we're done. So there's another session after this. So please go and support the early career researchers next door at the poster presentation. And then we have our dinner this evening at O Beach. So it's sort of in that direction from here. And that will be at 7 p.m. So we hope to see you all there. So thank you, but please go and support the poster session next door. I know we're a bit tired, but maybe just some fresh air out the balcony and around or through the doors here. So thank you so much.